Welcome to Learning Management System, Commissionerate of Collegiate Education, Vijayawada, Andhra Pradesh. Myself, Indravati, Assistant Professor in Biotechnology, KVR Government College for Women, Autonomous, Kurnool. Dear learners, the subject Recombinant DNA Technology is made easy for you. Do follow all my video lessons to cover different topics related to this subject. Dear learners, today we will be talking about plasmid vectors. In this session, you will be learning how plasmid is used as a vector in genetic engineering experiments. We will also be talking on natural plasmids and how they are differing from the constructed plasmid, and how the artificial plasmids like PBR322 and PUC series of vectors are used as efficient vectors in gene cloning experiments.
Let's study about plasmids in detail. These plasmids are the extra chromosomal DNA present in bacteria. So bacteria, apart from its main chromosome, which is represented in this red color, it will be also having small DNA molecules in many copies. They help in giving special characters to the bacterial cell. So these are closed covalent circular molecules. The size of this plasmid, it ranges from 2 to 5 kb, and they have the ability to replicate autonomously and have the inheritance property also. So in nature, different types of plasmids are present in bacteria. The first one is the F plasmid. So it contains fertility factors and promote conjugation in bacteria by the formation of sex pili. The second one is resistant plasmid, that is R plasmid, and it gives antibiotic resistant property to the host cells. The third one is col plasmid, and it encodes proteins called as bacteriocins or colicins. So they help in killing other bacterial cells, and only in that particular area the bacteria which are having this col plasmids, they will be flourishing its own population and kills other bacterial cells. The next one is degradative plasmid. So these plasmid provide the ability to degrade various type of organic and inorganic toxic substances which are harmful for the bacteria. Next, the last one is the virulence plasmid. This confers pathogenicity to the bacterium because it is containing virulence factors encoding genes. In this manner, in the nature, in bacteria cells, you will be having different types of plasmids, and they encode these type of special characters to the bacterial cell. Then how these plasmids they are used as vectors? Because we are saying that they are having the autonomous replication capacity, and they can generate high copy number. Copy number is nothing but the number of plasmids present in a bacterial cell. It's called as copy number. If there are more number is present, it's called as high copy number, and only less number of these plasmids are present, it is called as low copy number. So the copy number is decided by the nature of the replicon. If it is relaxed in nature, you will be getting high copy number. Then the small size of the plasmid. So it is preferred whenever the plasmid is of small size, it is easy to handle. And polylinker site. If the plasmid, it contains unique restriction sites for different types of restriction enzymes, so then that particular plasmid is preferred. Then marker genes, so they should be present in the plasmid so that you can easily detect the transform cell whenever you are inserting the foreign gene. The foreign gene is it successfully inserted or not, you can detect with the help of marker genes. And also the plasmid should have the ability to replicate in a large number of bacterial species, and it should have broad host range capacity. So like this different features which are necessary for an efficient vector, it should be satisfied by a plasmid. If all these features are exhibited, then that plasmid can be used as a vector in gene cloning experiment. But in the nature, that is in the natural plasmid, all the features which we have discussed, they are not present in a single plasmid. So that is for example, PSC101, it is a stringent and it is not relaxed. The replicon is not relaxed. So if it is there, then you will be getting low copy number. And for example, if you take COL-E1 plasmid, it is having poor marker genes. When marker genes are not there, then it is very difficult to screen the transform cell. And if you take RSF2124, the natural plasmid, it is too large in size. So it becomes very difficult to handle, to insert the gene or to recover the particular gene. So if the plasmid genome size is large, it is difficult to handle. So like this, all the features which we have discussed for an efficient vector are not present in the natural plasmid. So for this reason, artificial plasmids, they are designed and they are engineered by joining fragments taken up from the genomes of different organisms. So like this, the artificial plasmids like PBR322, then this PUC18 or 19 vectors and PMB322 vectors, these are the constructed plasmid or artificial plasmids or synthetic plasmids. So this will be having, that is the multiple cloning site or polylinker site is taken from one genome. Then replicon, that is a relaxed replicon. This portion of the DNA is taken from another genome. The marker genes, they are taken from another plasmid. Like that, different bits or fragments of the DNA are taken and they are joined to make a constructed vector, that is the plasmid vector, constructed plasmid vector. So this type of vector is called as artificial vector or the constructed vector. And let's study about this artificial plasmid PBR322 vector. So this particular plasmid was created in the year 1977 in the laboratory of Herbert Boyer at University of California, San Francisco. The PBR322, the P, it represents plasmid, B means Bolivar, R means Rodriguez. So Bolivar and Rodriguez are the scientists who constructed this plasmid, and 322, it represents the series number. So this PBR322, it is widely used in E. coli as cloning vector, and they are generally used to clone small size DNA fragments in the bacteria host cell. So if you see this plasmid, the size is 4,361 base pairs, and it is having a origin of replication which helps for the autonomous replication of the plasmid, and is containing a polylinker site containing nearly up to 20 restriction enzymes, unique restriction sites, and 2 marker gene. One is ampicillin resistant marker gene and tetracycline resistant marker genes. So in this manner, the plasmid PBR322 is having all the features which are necessary for a vector. So of this, the origin of replication portion, this region, it is taken from the plasmid PMB1, which helps in independent replication or autonomous replication of the plasmid. Then the marker gene, tetracycline resistant gene, it is derived from PSC101 plasmid. Then BLA gene, that is which is coding for ampicillin resistance, it is taken from transposon TN3. And like this, all these portions which are taken from the natural plasmids, they are combined to prepare the artificial vector PBR322. and you know that the size is approximately 4.3 kb, and these plasmid vectors, they are used to carry up to 15 kb of the foreign DNA. It can be inserted in this plasmid. Coming to the second type of vector, PUC 18 or 19 vector. So these plasmids, they are small, then they have high copy number, and the size of this plasmid is 2686 base pairs. It's of small size compared to that of the PBR 322. So this series of cloning vectors, they are designed in University of California. For this, U means University, C California, and P is the plasmid. So they were developed by Messing and co-workers in University of California, and these particular 18 and 19 vector, they just differ in the polylinker site orientation. In PUC 18 it is present in one orientation, whereas in PUC 19 the multiple cloning site is present in opposite orientation. That is the difference. And the marker genes which are present in PUC vector, they are LAC-Z sequence, and it is having nearly 13 restrictions enzyme, that is unique sites, they are present in the polylinker region, and it is also a broad host range spectrum is there. You can use this vector in different types of host cells. And the other thing to be noted is the origin of replication. It is derived from PBR 322, but it is having a single point mutation. So this particular PUC 18 or 19 vectors, they are lacking the ROP gene. That is, the ROP means repressor of primer gene, or is lacking because due to mutation. Due to this mutation, the PUC vectors, they are having a high copy number. The replicon is more relaxed, and you will be getting high copy number in this type of vectors. And the ampicillin resistant gene, it is derived from similar to that of the PBR 322, but it is having 2 point mutations. Then the LAC-Z gene is the one which is extracted from this E. coli LAC operon, and this encodes the enzyme beta galactosidase. So it helps in the easy screening of the transform cells. When you subject these cells, that is, whenever you plate these transform cells in a medium containing X-gal, they will be producing different types of fragments, that is, different types of colonies. We have seen the presence of two marker genes in the artificial vectors which we have discussed. One is the antibiotic marker gene, and another one is the LAC-Z marker gene. So in this slide we will see how we can select the transformed cells from the pool of non-transformed population. So here it is the media. The first one is a plate which is containing an antibiotic called ampicillin, and our transformed cells are resistant to antibiotic if our foreign gene is inserted in a portion other than the marker gene. The transformed cells are antibiotic resistance, and the non-transformed cells are susceptible to ampicillin. So in this manner, just when you plate these cells on a medium containing antibiotic, only our antibiotic resistant cells, that is the transformed cells, will be growing. And if you take the second example, that is the LAC-Z marker which is present in PUC vector, if you plate it on a medium, the cells are plated in a medium containing the substrate X-gal. The transformed cells, they are exhibiting one color, whereas the non-transformed cell, that is, you will be seeing two types of colonies, blue colored colonies and white color colonies. And by means of this, that is, the foreign gene is inserted in a portion other than this LAC-Z marker, then you will be able to see blue color colonies or the transformed cells, whereas the white color colonies, they represents the non-transformed cell. Just by the visual appearance, you will be able to select the transformed cells by means of this plating using antibiotic and substrate. Coming to the last slide, let's see the what are the points which we have covered. So we have discussed that plasmid is a closed covalent circular molecule which is of small size, and you will be having the capacity to replicate autonomously in the bacterial cells. The PBr322 plasmid, it is the size is of 4.3 kb, and it is able to insert up to 15 kb of the foreign DNA, and it is having two antibiotic marker genes, ampicillin and tetracycline resistant genes, and is having 20 unique sites for restriction enzyme. Whereas PUC19 plasmid, the size is 2.6 kb, and it can carry up to 10 kb of foreign DNA, and this plasmid is exhibiting high copy number, and two types of markers are present, ampicillin resistant gene and LAGZ gene. So this particular PUC plasmids, they have the, because of high copy number and this small size, they are more preferred in gene cloning experiments. With this, I conclude the session.
Hope you enjoyed learning this session. Last but not the least, I profusely thank Commissionerate of Collegiate Education for providing this LMS platform to reach a large number of enthusiastic learners like you. Thank you one and all.